computer. All right. So today, this is our great pleasure and honor to welcome Felipe de Brigard. Felipe uh, really embodies the kind of things we're trying to do in this network, the Philosophy in Biology and Medicine network. Uh, because, as many of you know already, Felipe is an associate professor of philosophy and an associate professor of psychology and neuroscience in the psychology and neuroscience department and the Center for Cognitive Neuroscience at Duke University in the US. He's a PI at the Imagination and Model Cognition Lab at Duke, and he works on memory and different faculties that are related to memory, including perception, imagination, attention, and consciousness. What I think is absolutely amazing is the way in which Felipe has managed to combine his interest in psychology and philosophy. And this has led to many interesting publications. Some of these publications are mentioned on our website, both very important papers, also books and anthologies. So, anthologies that put together some papers in neuroscience and philosophy. Today, Felipe agreed to present the paper I was mentioning before, namely a paper which is under review. So we really are grateful for that. I also very much like the fact that in the abstract and in the paper, Felipe criticizes de Brigard 2017. So it's a sort of self-critique, which is of course justified by the way in which papers are submitted, but also something which I thought was very, very interesting. So I really want to thank you, Felipe. I just want to mention one last thing, which is that during the Q&As, and I will mention that again, please say who you are in just a few seconds so that Felipe can know if you're a psychologist, a neuroscientist, or a philosopher, because of course the answer will depend a little bit on that. With that, I really want to thank you again, Felipe, and the floor is yours.
All right, thank you very much for inviting me. Let me just make sure that I can share my screen. And let's see. All right, can you see my presentation
That's
here?
perfect. We see the full screen. Perfect.
Okay, wonderful. So thank you very much again, Thomas, for inviting me to give this talk. So I'll start off with a couple of caveats. The first one is I am both a philosopher, but I'm also a cognitive neuroscientist. And one thing that has happened throughout my life and throughout my career is that there are moments in which I do those two things kind of independently of one another, and there are moments in which they sort of intersect. And sometimes I just don't know when they are going to do that. So the story of this paper is interesting. I was asked to write a paper years ago on this special issue on the notion of cognitive system. And I have had a beef with or rather try to understand what a cognitive system is. And I always assume that people think that, they talk about what cognitive systems are, but I think that there are always sort of questions as to exactly how to demarcate cognitive systems. And I was very interested in this question back when I was doing my PhD. And so I thought, well, I had a little bit of an argument in a philosophical argument against one very sort of famous proposal for what a cognitive system is. So I wrote that paper, which got published almost, like 8 years ago or so. But then it was unsatisfactory, the solution that I gave in that paper. And in the interim, in the last 8 years or so, and I think that Daniel, who's here in the audience, knows about this, I have gotten really interested in topological models of fMRI data. So my lab does some fMRI, and we have done a lot of sort of new kinds of analysis using topological formalisms to try to understand, try to interrogate neural systems differently. So I decided to sort of go back to that old paper of mine and try to sort of give a more satisfactory solution to my own question, which is why I was sort of responding to myself and why I write in the third person in this paper. So hopefully then the story will be clear. And one last thing that I would say is there's a little bit of math in this talk. I will go slowly through it, But if you have the paper, you might want to follow along. The talk is just basically a presentation of the paper. All right, so without further ado, let me just start. Uh, so as I mentioned, one of the main goals in cognitive neuroscience is to try to identify neural structure. I think that someone is unmuted. I think so. Again, as I mentioned, the main goal of the cognitive neuroscience is to identify neural structures that correspond to the cognitive systems that are identified by our best psychological theories. Unfortunately, this goal faces many conceptual difficulties, and one of those conceptual difficulties is to try to understand what a cognitive system is. So a traditional view in philosophy is to define a cognitive system according to what I call an agent relative view. This agent relative view is sort of a top-down view. It starts off with the idea that you have an agent, and then you decompose that agent into smaller and smaller and smaller and smaller modules. For philosophers in the audience, this might come as you might recognize this as the kind of strategy that was advocated by Dennett in 1978, by Bill Lycan in cognitive science. This very same sort of functional decomposition has been advocated by lots of people, Simon, Marsh, Alice, and so forth. And in fact, more recently, I guess 25 years ago, Cummings suggested that the process of explaining cognitive faculties via breaking them down into its component system is exactly what the psychologist is supposed to be doing. So basically, the idea is something like that. This top-down approach of functional decomposition is you start with an agent, right? And then you subdivide that agent into a system, and then a subsystem, and then another subsystem, and then another subsystem, until you finally bottom down at a level in which there is no further functional decomposition. I like to say that this is the level in which behaviorism is true. You don't subdivide that system into any more subsystems. But as some of you might know, this approach was sort of very like it was challenged by what is sometimes known as the extended cognition approach, which is the idea that look, we assume that the limits of a cognitive agent are identical to the person, but it turns out that what an agent is can go above or outside of the skull. So the notion of cognitive agent cannot be taken for granted. So instead, what they suggest is to try to understand what an agent is, because agents could be sort of these amalgams of people with objects or people with the environment and so forth. So instead of starting from a notion of cognitive agent, we should take a different approach. And so that's people call this approach the bottom-up approach, which consists in finding the necessary and sufficient conditions for a certain component to be a constitutive member of a cognitive system, or for a set of components to constitute a cognitive system, as opposed to either a non-cognitive system or a non-system. This approach comes with a challenge as well, which is the problem of trying to identify a reliable way to determine which of the mechanisms that are causally responsible for the production of a certain cognitive process end up constituting a cognitive system which is responsible for such process, and which ones do not. So for the philosophers in the audience, you might recognize this as a debate that happened, in the early 2000s, and that gave rise to a very influential proposal by Robert Rupert in 2009. According to this proposal, a cognitive system is constituted by those mechanisms or those capacities, abilities, etc., that differentially contribute to a specific task and that are integrated. And this notion of integration is critical for Rupert's account. Which mechanisms are integrated into a cognitive system depends, according to Rupert, on their conditional probability to cause or to contribute to causally contribute to the target cognitive task relative to any other causally contributed mechanisms. So how do we know that? The general idea is, how do we know that a particular, say, brain mechanism contributes to a particular task, and it is part of the cognitive system for that task? Well, it has to do with the frequency of occurrence of the cognitive mechanism in bringing about that particular task. I will get to that in just a second. So this is the context in which this debate is happening. And as I mentioned almost a decade ago, I wrote a paper in which I tried to put pressure on Rupert's account by basically showing that this account is interesting, but it fails to take into account the fact that brains change over time, that we as biological creatures are constantly changing. And by constantly changing, you can play a little bit with the frequency of use of these different mechanisms. And then I argue that the notion of cognitive systems a la Rupert breaks down when we take time into account. Specifically, what I argue is in my challenge to Rupert is that a successful account of a cognitive system for cognitive neuroscience needs to be sensitive to the fact that neural mechanisms change over time. I call this diachronic dynamicity, that is to say, they're dynamic over time, and yet often they don't change their function. So they have what I call diachronic dynamicity, but they also exhibit what I call functional stability. And that was the paper. At the end, I ended up suggesting, said, well, there might be some alternatives here, but it was just very hand wavy. It was mostly sort of a negative paper against Rupert. And I just didn't have a solution back then. So what I want to do in this talk is to try to offer a solution to the very same challenge that I offered against Rupert. It will turn out to be, and I have, and this is incidental, but Robert and I have talked a bunch since I wrote this paper, and it turns out that I don't think that he's completely against the particular view that I am going to be suggesting here. So anyway, so that's the context. So to give an answer to that challenge, I want to first talk a little bit about the challenge in itself and why I think that the fact that our brains change over time and exhibit this diachronic dynamicity with functional stability is a challenge to any account of cognitive system that is static. Then in section 3, I am going to move toward a dynamic account of cognitive systems. And I am going to use in particular a topological framework that is known as multi-scale network. I will tell you a little bit about how these multi-scale networks work in cognitive neuroscience. And then I'm going to introduce a problem and a solution. The problem is how can we include time as a parameter in these multi-scale networks? And then the solution, which is what I offer, a way of parameterizing time in these multi-scale networks, might lead us to think of about a possible way in which we can actually end up with dynamic cognitive systems. And finally, I end up with a few objections and some final thoughts. So that's the plan. We already finished with the introduction. So let's go to the second part, static cognitive systems and the changing brain. So as I mentioned, the strategy that Rupert has to characterize cognitive systems involves basically two general steps. The first step consists in ranking all the sets of mechanisms that have ever contributed to bringing about a particular cognitive task based on their frequency of co-occurrence, and then to find a suitable statistical threshold below which every set is removed. And the second part is to record the frequency of occurrence of each mechanism in the remaining sets, and then identify a suitable statistical threshold such that any set below the threshold is removed. So this is a lot of information, so I'm just going to use a toy example. Oh, and then the idea is that the members of this resultant set are integrated and that they end up constituting the cognitive system of the relevant task. So this is a sort of a toy example. Suppose that through time, obviously this is going to be a much larger number, but suppose that through time, there's 6 different brain mechanisms, M1 to M sub 6, that have causally contributed to a particular task, called that task T. So the first step consists in ranking in a list all the subsets of those 6 mechanisms that have ever contributed to T, and see, the first time that suppose that I did the task T at time T1, and when I did it, then the mechanisms M1, M2, M3, M4, and M5 were engaged, called that subset M1, capital M sub 1. But now suppose that I did that task at another time, at time 2. And now when I did that task at time 2, it was brought about by this other subset of mechanisms, M1, M2, M3, and M6. So notice here that I don't have M4 and M5. So call that subset M2. Do the same for the next time that you do T, the next time that you do T, the next time that you do T, right? So that's how you do it. So you collect every time that you do T, then you collect all the subsets of those mechanisms that were engaged. And then the next step consists in assigning a probability, and that probability is based on the relative frequency of each one of those mechanisms in bringing about T. So again, each probability is conditional to every other mechanism and every other subset of mechanisms within each of the big M sets. So for instance, we assign the probability to M1, given that M2, little m1, given that little m2 was active, to 0.9. That is to say, it is almost always the case that when m2 is active, m1 is active. Almost true too for M3. It's almost always the case that M1 is active when M3, if given that M3 is active, not as much as given that M2 is active. And then you continue doing that with every combination of frequency of use of each one of the sub-mechanisms, and then you rank them given their probabilities, right? So as you can see, I used these 3 dots to sort of indicate that anything above that is going to be above chance, and anything below that is going to be below chance. Now, chance is also going to be determined on, on a case-by-case basis, but here a very simple example is just simply to use 0.5 as chance. So you basically take all of those mechanisms that are above that threshold, suppose that their threshold is 0.5, and then you don't include the ones that are below that threshold. And then the final point is to identify the set of all those sub-Ms, or all those mechanisms, which given the frequency of co-contribution is more likely to bring about T. So in other words, once you have ranked every probability for every single mechanism to bringing about a particular task, given every other mechanism that is involved and every other subset of mechanisms that are involved, you find the set of all those mechanisms whose combined probability is above this threshold. And according to this identifies one specific set of mechanisms whose history of co-contribution in bringing about T is above chance. So for instance, in our example, it might be possible that the set MT, which includes M1, M2, and M3, are the mechanisms that are much more likely to co-contribute to bringing about T, given all other mechanisms that at any point have contributed to bringing about T. So let's assume that that the probability of bringing about T, given that this set of that we call here MT, hopefully you see my cursor, given that that the probability of bringing about T, given this set is something like 0.78, that's the higher probability, given again that if you sum all the probabilities of all of these mechanisms and all of the subsets of all those mechanisms, this is the highest probability set. That is basically how you are done. MT, according to this, that subset, 1, 2, and 3, forms an integrated set, and it is to be identified with the cognitive system for that task, as opposed to mere resources used in the process of bringing about T. Now, if you are familiar with this debate, you know that the reason why Rupert brings about this criteria is to rule out things like an iPhone or a book, like in cases like the Clark and Chalmers case, in which Otto is supposed to have this external pad where he has this information, and the idea is, look, this pad really very infrequently co-occurred in Otto's mental life in bringing about this particular task, and as a result, it should not be included as part of a cognitive system in Otto that brings about T. So that's what this criteria was introduced, to rule out external things that we employ to bring about T, but that do not constitute a cognitive system, that is to say, that are not integrated in the cognitive system to bring about T. That is the nature of the debate. Now, the problem that I pointed out is that since this strategy is so dependent on the relative frequency of co-contribution of causally relevant mechanisms, it is also highly sensitive to diachronic change. And the problem is that we, as we age, our brains undergo constant change. There are lots of changes that are constantly going on at different temporal scales. Dendritic branching and pruning, changes in dopaminergic expression, increases in white matter hyperintensity, volumetric reductions in the cortical mantle, what have you. There are lots and lots of different changes. But what is interesting is that despite the fact that we have all these changes in our mechanisms, often we don't see a change in performance. That's the functional stability. Here are two very clear examples from the aging literature that I discussed. One of them is known as the Harold model, and it was discovered in 2002 by my now colleague and previously Professor Roberto Cabeza, and it has been replicated over and over and over again in all sorts of neuroimaging studies, including this one is PET, so including fMRI and PET and AMEG and so forth. Basically, what the result shows is that when we are young, we tend to recruit one hemisphere in doing a particular task more so than the other. However, as we age, that very same task tend to recruit both hemispheres to a greater extent. So that's what they call it, hemispheric asymmetry reduction in the old, right? So in other words, when you're young, you tend to do one task and recruit typically only one hemisphere, but as you're old, then you see that you recruit both hemispheres for the same task. This was in their paper, a review showing this reduction in asymmetry between hemispheres as a function of age. So that's one change. You do the same task. There's no difference in performance, and yet it looks as though the cognitive, the neural mechanisms that are engaged are different. Here's another phenomenon that also occurs during aging. Is known as the PASA model, which shows that in addition to this reduction in hemispheric asymmetry, there is also a back to front pass. In other words, when you're young, here is the YP, is the young adults. When you're young, when you're doing a particular task, for instance, in that task, you tend to over-recruit the occipital cortex, but you definitely don't recruit much of the prefrontal cortex. However, when you are old, you tend to over-recruit that occipital cortex way more so than young, and under-recruit that occipital cortex. So there is more recruitment in frontal areas and less recruitment in occipital areas, even though, again, there is no change in performance. So how would you handle, if you were Rupert, that kind of diachronic change from young and old in their account of cognitive systems? So here is, again, our toy example. Suppose that we have an experiment that is a phase 1 experiment in which we test, one particular task, and we notice that for this phase, suppose this when I am 16 years of age, there are these 4 mechanisms that end up being integrated, and as a result, we call them the system or the cognitive system for that task. But suppose that that very same individual, me at age 70, does the exact same task. There is no change in performance, and yet what we find is that there is a change in that subset of that ends up being included here in the cognitive system. So which one of these is the cognitive system for task T for me? Is this the one that I used when I was 16 years of age, or is the one that I use when I am 70? That's the question. So, well, this is another way of asking the question: is this the set that constitutes the cognitive system, or is this the set that constitutes the cognitive system? So, Rupert has a number of possible alternatives. So, one possibility is to say that the cognitive system for T is the intersection of MT and MT star. So, neither M2, right, nor M5 would be part of it. I think that this should have been M3. Well, I think I made a mistake in that in this, but in the paper is the correct one. Another possibility is to say that since S used MT much more so that empty asterisk, right? You just use this for like 60 years, whereas this one has been only used, say, for 10 years. Then this one is the cognitive system instead of MT. The problem with that alternative is that it makes a cognitive system highly dependent on our age and our lifespan. Suppose that there's, our medicine evolves in such a way that we manage now to live 200 years. And it turns out that because we live 200 years, now MT was used for 60 years, but now MT star would have been used for, 120 years. So now it becomes MT star, the ones that is more frequently used relative to MT. And finally, another possibility is to say, look, we're talking about two different cognitive systems. One is the cognitive system for MT when S is young, and another one is the cognitive system for MT as of MT asterisk for when S is old. Well, the problem with that alternative, though, is that the only thing that we have kept constant is T, and T is determined by the performance. That is to say, the task is determined by their performance. So in changing and saying that there are two different cognitive systems, it will be really hard to explain why is it that we have two cognitive systems when it is exactly the same task and exactly the same performance. So that is the challenge. The challenge is how can you incorporate these dynamic changes in the brain when you try to give an account of a cognitive system. Hopefully that is clear. So I in the paper, I mentioned this. I said that, to be fair, in a recent paper, Rupert in 2019 defended his conditional probability of contribution account against my criticisms. However, my interest here is not so much to go back and sort of respond to Rupert, but rather I want to do is offer a different strategy to tackle the challenge presented by me in 2017. Incidentally, that challenge has apparently there are other people that are discussing that challenge, because I just saw a recent update in the Stanford Encyclopedia of Philosophy of Neuroscience, and it turns out that there are more people that have been trying to tackle that challenge. I have to read them. So anyways, what I want to do more precisely is to offer an alternative to Rupert's conditional probability of co-contribution approach that can help us to identify cognitive systems for specific tasks in a way that preserves both their diachronic dynamicity and the functional stability. So from the previous discussion, I would suggest that a couple of lessons. The first one is that attempting to identify the cognitive system for a particular process or task in a way that is both stable and dependent on the frequency of engagement of its component mechanisms might be insufficient to give us an account of a cognitive system that changes through time, or that supports changes through time. And the second lesson is that we should probably find strategies that allows us to parameterize time across different individuals or across different times, independently of the mechanism's frequency of engagement. So how can we parameterize time independently of the mechanisms of frequency? So the account that I want to suggest builds upon what is known as multi-scale networks. So let's start thinking about our brains, as massive networks that have differential elements or nodes that are connected by pairwise relationships or edges, right? So many of you are probably familiar with this kind of way of thinking about brains. Now, what constitutes a node or an edge is going to depend, among many things, on the particular scale of analysis. Now, in the case of brains, there are at least three scales that interest us. First, there is a spatial scale that goes from the very micro, to the very macro, right? So from such, networks are going to include things like neurons and nodes at the micro level and synaptic connections. But at the macro level, they're going to include, for instance, discrete parcellations of brain tissue or areas and so forth. Second, brain networks also vary along a topological scale that grows from very local networks, right, to very general or global networks. So that's the second dimension. So we have the spatial dimensions, and we have the second dimension, topological. And finally, and critical for our purposes, we can also find networks that vary along a temporal scale. So we can find different network organizations that just vary in the order of seconds, but we can also have the order of sort of minutes or days, and even we can have changes in network organization that happens through evolution, right? So like through, years and centuries and so forth. So these are three dimensions across which we can think of brains as networks vary. Now, given its multi-scale sort of complexity, identifying structures in the brain is very tricky. Thankfully, the recent strategies for detection of communities, that is to say, subnetworks that are internally dense and externally sparse, that have shown to succeed in detecting structures at different scales. So although there are a few community detection strategies in the offing, one of the preferred strategies is what is known as modularity maximization. And it was developed in full by Newman about 20 years ago. What modularity maximization does is that it partitions networks to maximize the measure of modularity, which basically tests the observed patterns of connections against the pattern that would be expected under some null model. In other words, when you run null models in which you would say like each node has the exact same probability of being connected with other node, that's the test network. You see if the brain, for instance, has a organization that is significantly different from what you would expect at chance. And what the Q modularity algorithm is that it maximizes this selection of these communities that are more statistically significant from the null hypothesis, namely that, and the null being that each node has the exact same probability of being connected with other node. And in doing this, what you end up finding is that there are sort of nodes that are really, really sort of interconnected with each other and less or more sparsely connected with other nodes. And this forms like these kinds of little communities or modules, as they are called. Now, the problem is that Q modularity is often level dependent, right? So you say, okay, I'm going to find community Q modularity just at the level of the cortical parcel, or I'm going to find Q modularity just at the level of this column in this part of the cortex, and so forth. So to detect communalities across scales, we need to identify a parameter that is introduced so that it can maximize not only within one particular level, as you want within one at the micro level or the macro level, but also that it could maximize the selection across a topological scale. So if you go back to this one, we can maximize the selection of communities or modules at different levels. But remember that we want to now be able to identify them at different spatial and also a different topological degree. So we're talking about these two dimensions, spatial and topological dimensions. So this is where it gets a little mathy. I promise I only have two formulas this is one of them. So this is the way in which modularity has been augmented to include what is known as the gamma parameter or the resolution parameter. I will explain this in just a second. So here, just for those who are interested in the math, here A indicates the observed weight, which is the strength of connectivity between two nodes. So I and J are nodes, and this says that they're really, really tightly connected or not so tightly connected. And then then this also shows that is the expected connection under the null hypothesis. So if this one is like super duper higher than what you would expect under the null, it's going to give you a really high number here, and the opposite if it is not too different from what you expect from under the null. And this is the resolution parameter, which is the way in which you can detect these structures across different scales. And finally, there is this funny uh, parameter here is called Kroneker's delta. The Kroneker's delta function basically assures that the contribution to Q, to the modularity, comes only from these two guys by rewarding assignments of these two modules as opposed to any other module. This is a very important point because at any point, any module, any node could be communicated with any other node. But you really want to say, like, if there is just sort of accidental correlation between one node and another node, we don't want to include that accidental connection. We just want to include the two nodes that are of interest here. So this delta just basically penalizes any other node that is not I or J. That's just a way of making sure that you're just looking at each independent part of nodes as opposed to any possible connections with other nodes. All right. So using multi-scale modularity detection, they have been used in the last few years by some groups, in particular Danny Bassett's group. So what they have shown, for instance, is that you can identify structures at different levels at different, not only topological levels, but also at different levels of space, right? So you can have a very broad resolution parameter. This is this gamma parameter here. You can have a very broad resolution parameter, and then you show community structures that are pretty big, as you can see here. But within those community structures, you can have that resolution parameter being a little bit smaller, and then you can find communities within larger communities that are also pretty stable. So here is a way of seeing this. When your resolution parameter is pretty big, these are these big squares, you can see lots of patterns of interconnectedness. But you can also modulate that resolution parameter to get more interconnectedness at lower levels, lower physical levels of description. And then you end up seeing that there are these smaller structures within the larger ones and smaller within the larger ones. So now you're getting this resolution that we were looking for before. So the bottom line is that this is a promising strategy to identify hierarchical structures along spatial and topological scales for neural networks that are associated with specific cognitive tasks or specific processes. The question now, the critical question now, I hope this is clear how you can sort of have this structure that are hierarchically organized, and it tells you, look, it's like, if you think about it, it's like in a city. In a city, you can have neighborhoods, and within neighborhoods, you can have communities of houses organized, and so forth, or many, you can see it in that specific way. And it's the interaction between those neighborhoods and the municipalities and counties and so forth. This is going to vary, of course, from country to country. But it is this structural organization is what we are basically trying to identify in the brain. The question is, so far, I have only talked about the topological dimension and the spatial dimension. So now, can we include a temporal scale as a parameter in multi-scale community detection strategy in order to capture not only variation due to spatial and topological resolution, but also due to change over time? Well, I think we can. All we have to do is to start thinking about slices, not only as being bigger or this network identification, not only as being bigger or smaller in terms of space, but also we can think of them as varying through time. So we can think about each temporal, each community detection resolution as being at a particular time, call that a slice, a temporal slice, and then you can see how they behave through time. This is and this is, I promise, this is the last last formula. So this is a way of augmenting the modularity formula with a temporal parameter. So now this is very similar to a formula that we had before. As you might recall, we had A was the strength of the connection between two nodes. But now we are using two nodes and we are indexing to each particular slice. Again, the P is what you would expect under the null hypothesis. And this gamma, again, is the resolution parameter. Then there is also a coupling strength, and this coupling strength is the one that is going to end up parameterizing time. The W is parameterizing time. How does parameterize time? It tells you what is the strength I'm going to show. It tells you what is the strength of connection, not within the same slice, but between, say, this node and 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 the next iteration of that node in the next temporal slice. I'm going to use a toy example in just a second, but just so you know how in the math it looks, this is the coupling strength. And I'm going to sort of skim over the other ones because what matters here is this omega definition. Basically, what this does is just a normalizing parameter, but it doesn't matter too much for our purposes. For our purposes, is this W parameter is a way of parameterizing time. Now, there are a number of advantages to employing multi-scale temporal networks. Oh, and by the way, what I am doing here is following the math that was developed by Mucha et al. In this very important paper that came out in Science about 15 years ago. So one advantage of this multi-scale temporal network is that it is agnostic about the temporal scale. So that W parameter, that omega parameter that I mentioned could be TRs for fMRI, it could be seconds, could be years, what have you. Now, of course, if it is years, the parameter is going to be very tricky to do. The longer the time, the more you have to take into account, for instance, the fact that that brains decay over time and so forth. So the parameter is going to change. This is one of the things that is the trickiest. We'll get to that in the Q&A if you're interested. A second advantage is that estimates of community structures across topological and it allows us to estimate community structures across topological, spatial, and temporal scales. And in addition to that, introduces new measures. They call these measures of flow for community across slices. I'm going to give you one example. To my knowledge, the first use of temporal networks in fMRI or in cognitive neuroscience was done by Danny Bassett and colleagues when they were, I think, a graduate student or postdoc with Scott Grafton. What they show is that when you take into account changes that occurred through time as a function of learning, so this was a task in which participants were learning something while they were in the scanner, what they were looking at was changes in these community detections that occurred through time. And what they sort of discover is that nodes that were highly flexible, and they defined flexibility as the number of times that a node changes modules through time. In other words, if a node is in one module at a temporal slice number 1, and in a different module at a temporal slice number 2, and a different module and temporal slice 3, that number, that parameter that they call flexibility, actually predicts how well you learn the task. And this is really cool. As any of you who are interested in topological measures knows, when you find properties of a topological model that are highly predictive, then it is really cool. And this is exactly what we find. We find that there is a temporally indexed topological property that is very good at predicting learning. Ever since similar, they haven't been used too much, I should say. Like if you do temporally parameterize multi-scale networks and you do a search, there isn't a whole lot of papers, but some of them have shown these dynamic reconfigurations, and then they show that certain measures of flow are predictive of certain things. Like for instance, surprise. I think that this one, positive affect, surprise, and so forth, are predicted by this network flexibility. So as I mentioned, although just a handful of studies have employed multi-scale networks to detect these communities across time, my contention is that they can become very useful strategies to identify stable topological properties across all sorts of dynamic multi-layered networks. So I'm going to end up this presentation with an example that I think can be generalized. So this is a paper from Bassett and colleagues from 2013, in which they show that during skill learning, there is a set of core regions, right? Here is a set of core regions that have shown that during, mainly comprising these sensory motor regions. As you can see, this is the core regions, motor control and visual cortex in particular, which are mainly multimodal association regions that tend to be sparser, than the periphery. So let me just recap here. There's a set of core regions that are mainly motor and visual, and there is also a set of more peripheral regions that are mainly multimodal, right? My guess is that the identification of topological properties of multi-layer networks that are more or less stable across time could be applied in a similar fashion here. So what we see here is that these core regions that tend to be more stable end up being something like the system for visual motor learning, right? By contrast, these other regions that are more association are, and as a result, that are not part of this modular organization that is core through time, they're going to be more like the other parts that are active when you are realizing a particular task, but that you wouldn't call integrated. So now you see how this is starting to link back to the Rupert discussion. So the idea is when you use this multi-layer temporal networks, you end up finding that some modules are highly stable across time, whereas other ones are less stable across time. And when you look at which modules end up being, it turns out that they're the modules that you would expect to see for tasks that are, say, in this case, visual motor learning, whereas the other ones that are active, perhaps even causally responsible, but less stable across time, they are going to not be part of the cognitive system. That's my proposal anyways. So let's go back to the cognitive systems. So we have a question. We ended up in the previous part with a question as to whether this .m2 or m5 were part or not of the cognitive system for T. And I offered three possible solutions. The intersection, that just basically saying that neither of them is part of the system, or just basically saying that they're two different tasks. Well, I want to suggest a different solution based on temporal multilayer networks. The answer, I was going to argue, is going to depend on whether such nodes end up being part of the dynamic multilayer network that underlies T. So here is a graphical depiction of what I am saying. Suppose instead of thinking about this in brain space, think of this in topological space. Suppose that at a first temporal slice, the nodes that are active, the mechanisms, if you want, think of each node as a mechanism, that are active are, these ones, and form these community detections, and the green one, the red one, and the blue one. And think about each one of these nodes. I just numbered some of these nodes: M6, M2, and M5, and M1. So think about M1. M1 is definitely going to be part of it. It's super stable. These dotted lines are that omega parameter that I was talking about. So if you think about the fact that M2 is correlated with in S2, in S3, in S4, that omega parameter that I was talking about here is going to be super duper high. And as a result, that M1 is definitely going to end up in the queue in this multi-layered temporal module. Now, take a look at M2. M2 is not included in a module here. It is included in a module here. It is not included in a module here, and it is included in a module and it is included in a different module here. So M2 is absolutely not stable at all, right? It might have been somewhat coactive in certain temporal moments, but it's definitely not stable, right? Now look at M5. M5 is rather stable, right? So it's like M1. And M6 is not part of any module. It might have been active while engaging in those tasks, but it was never part of any module. So according to this, M1 and M5 right here are going to be part of the same of the cognitive mechanism for Q because they're part of a resolution of the multi-layer network. And M6 and M2 are not going to be part of that. This one because it was never picked up by any module, and this one because it was picked up by two different modules and it was not stable across time. Hopefully that was clear. So the take-home message is that multi-scale temporal networks offer a useful way to characterize neural structures engaged in a specific task in a way that preserves both functional stability and diachronic dynamicity. The temporal agnosticism of the coupling strength parameter, the omega parameter, enables us to use multi-scale temporal networks at different temporal scales. And in principle, I argue it should allow us to identify modular alliances or groups that are integrated across tasks, tapping at putatively similar cognitive processes. This is my last slide. I'm going to quickly end up with some objections. So one objection someone might, and these are objections that were raised to me the only time in which I presented this before in public. So one objection is to say like, look, Felipe, what you're doing is super cool, but networks are not explanatory. They're merely descriptive. What you're doing is merely descriptive. They're not telling us why is it that, these networks are producing T. They're just telling us that these networks are producing T. To that I have, and many of you, if you're philosophers in neuroscience, you're probably aware of this discussion that's going on as to whether topological networks are explanatory or not in cognitive neuroscience. So there are two possible answers here. One answer is to say, I don't care about the debate about explanation versus description. My project is descriptive. I am just trying to describe what are the cognitive systems that underlie a particular task. However, there is another solution, and this is what my former postdoc, Trey Boone, pushed me to do. This is like, look, you can say more about this. You can say, actually, the people that say that networks are not explanatory are wrong. Actually, networks can be explanatory. So, of course, this is a burden shifting kind of argument. I will have to say more about why I think that topological networks or network models are explanatory cognitive neuroscience. And I will have those things to say in a couple of forthcoming papers precisely with Trey Boone. So that's one objection. Another objection is to say, uh, great. So you're talking about nodes, but the original debate was about mechanisms, and nodes and mechanisms are not the same. This is a fair point. Nodes are a matter of taste, and in topological network models, what constitutes a node is something that's a little bit of a matter of taste. People decide what a node is on the basis of their algorithms and the resolution of their instruments and so forth. It turns out, and I can say more in the Q&A, but it turns out that many of the approaches that are employed by the people in the literature end up with convergent solutions for nodes. So you might end up with one instrument saying these are the nodes, and another instrument these are the nodes, and often they end up converging, suggesting that there might be an underlying biological structure that the node corresponds to. So my suggestion is let's go pragmatists. Let's continue using nodes as long as they're useful to give us these measures of flow that are predictive of behavior and so on and so forth. And then as we collect more data, we might approximate nodes, the right resolution for us to identify these multi-level scales. I mean, these multi-level networks. And finally, someone might say, yeah, but we're talking about cognitive systems for cognitive processes. And all you are doing is giving us cognitive systems that are indexed to particular tasks. And tasks are not the same as cognitive processes. And to that, I would say, here. I completely agree. We just published a recent paper in which we argue, among other things, that we measure tasks, we don't measure directly cognitive processes. This is a very well-known point in psychology and in cognitive neuroscience. But the thing is that that's still important. We can still talk about cognitive systems that are engaged in tasks, and then we can do some extra work as to see how those tasks, index different cognitive systems. I mean, different cognitive processes. So it is still linking it to task is a first step to see how they are linked to cognitive process. More work is required to know exactly how that task indexes a particular cognitive process, but it's still a first critical step to get to that point. And with that, I want to thank you for listening to this talk.
Thank you so much, Felipe. That was great. And we have time for questions. So one thing I said before, but I will say again, is that I would suggest that you introduce yourself by just saying your name and whether you're a philosopher, cognitive scientist, psychologist, whatever. That I think is going to be helpful. Um, I don't see any questions for now. I have one question which is very naive, but which is connected to what you said about Rupert. I mean, in my view, Rupert could say, yeah, I'm perfectly fine with what you said. It's just that you're adding this dimension of temporality and this dimension of diachronic dynamicity. And if I were to introduce that kind of temporal questions, I would do exactly the kind of things that you just suggested. Maybe not, but I'm just asking here, is in the end not going to be the case that what you presented today is in fact to a certain extent orthogonal to the initial. I mean, it's just adding something different to the initial suggestion of Robert. Am I right here or am I wrong?
Uh, well, that's not exactly what Rupert said to me or what he says in his 2019 response. What he says is that I mean, and one thing that I should mention is that this notion of integration is is explained in two paragraphs in his book, right? So I am really picking on just two paragraphs of his book. The problem is that his notion of integration makes is very important in the book. He dedicates just a tiny little bit to spelling it out, and as he himself acknowledged, he wasn't the clearest with it, and he didn't think about all the possible consequences and so forth. Uh, so what he ends up suggesting the in his response is that he wasn't thinking of each cognitive system as a subcomponent of the agent. That his that he wasn't clear enough in his presentation, and that he sort of saw why is it that I thought about this as a way of thinking of a cognitive system as each one of those circles, these concentric circles that I had there. And he said that was not the view that he had. And then ultimately, as you say, he because what ends up happening is that he says, look, this is just a pragmatic sort of task of trying to determine for one specific task, but I wasn't saying that these mechanisms are just locked into this mechanism in the brain is whenever it's used is going to either participate or not participate on this particular task. It's just like this is one way of determining for one specific task, but I am not committed to this being a cognitive architecture structure. So that's sort of his solution, or what he had to say. And I think that he would say, yes, if we were something like, if we were to apply the frequency of co-contribution approach to identify cognitive architectures, the way in which I explained at the beginning, then we will have to parameterize time. And if I was doing that, which I'm not, but if I was doing that, then I would parameterize time. And the last time he and I talked, he said, like, I think that your solution is probably very adequate. So ultimately, we are not, this is not your traditional philosophy talk in which he said, I said, like someone else said, but it's more like this is something that should be clarified, and this is one strategy to clarify it, and we end up being friends at the end.
Okay, good, good, and yeah, good to have that confirmation. Maël,
Yeah, hello, can you hear me?
Yes. Hi,
Okay.
Mile.
Hello. Thank you very much for the talk. So I'm Maël Lemoine, I'm a philosopher of science. The question I wanted to ask, so I'm not sure I got because you lost me by the end of the talk, so I'm not sure I got everything, but you will explain.
Yeah.
So it's a basic, I would say it's a basic experimental question. So when you try to map a task, a specific task on the brain, and so you said it changes with time. Okay, I get the point. But how can you be sure that what looks like the same task is in fact the same task? Because it could be, I mean, I'm thinking simply of, I mean, the brain is doing many different things at the same time. So in particular, even in the same area, I suppose. So
Mm.
how could you know that, like, for instance, I don't know if you're doing a multiplication, for instance, so that's one task. But at the same time, to perform that multiplication, in a young brain, you need to do a lot more at the same time, while in an old brain, maybe you need to do a lot of other things to do the same thing. So, what about? I mean, I mean, that would be a way of salvaging the most intuitive explanation, which would be that the task is localized, but it's so small in everything that you observe that you don't notice really what. So you get my point, I suppose.
Yeah, I absolutely get your point, and it's a very good point. It's very well taken. So, one so we think that it is the same task just based on performance, right? So think about one very well used task and a very well known task. In fact, one of the tasks that uses this temporal parameter, which is the end back task. So the end back task is widely used in cognitive science and in psychology. Has been used for like 70 years. It consists on showing, say, letters and one after the other, right? And I tell you, whenever you see the letter F, I want you to tell me what was one slide before, 2 slides before, 3 slides before. That is called an end back task. And the more n, the more the number is of slides prior, the harder it gets. And it is supposed to be a task that measures executive control or working memory, depends on who you ask. And the idea is that it's a very simple task. It's not like multiplication, because of course in multiplication, my daughter is 9 years old and she multiplies things in ways that are very different from how I do it, because now they teach math differently in this than the way in which I learn it. But suppose that the task is something like n back, which is very, very simple. And then the idea is that anyone that can sort of read, or you don't even have to read, you can just have symbols, will respond it. So we find those differences so for instance, the asymmetric reduction from instead just only recruiting one hemisphere, you recruit both. You find that reduction with tasks as basic as that one. So the way in which cognitive neuroscientists interpret that, and again, there is no difference in performance. A 16-year-old or a 24-year-old will perform just as fine as a 70-year-old in this task. So I would argue, is the same task. If you ask them, did you do anything different? They don't. There is no introspection, the fact that they are going to do it different. But the aging people, or rather the people that work on aging, not the aging people that we work on aging, argue that the way in which we should interpret the brain results is by saying that the brain employs different strategies. I personally don't like that solution, because it's kind of like we don't have any other evidence rather than differences in brain activation to the point that they are applying different strategies. You ask them, how did you do it? And they tell you, well, I just remember what was before. They say exactly the same thing, whether they're 24 or 74, right? Their performance is identical. There is no difference, right? So the notion of strategy is just another way of saying, well, the brain seems to be behaving differently. But it's not that they're doing we don't have reason to believe that they're doing a different cognitive strategy, right? So that will be my response to that. Is like, and if we don't have any reason to believe it's a cognitive strategy, if we have every reason to believe that it's the same task because of behavior, then we have this conundrum as to how to explain the change in brain activation.
So if I can clarify a little bit my question. I mean, so I get your point, and I think I would agree with you on the end back task, of course. But what I meant is that, for instance, to perform the same task, maybe you need to inhibit, for instance, other processes at the same time, and that might explain the difference. Like, for instance, in a young brain, you will need to inhibit certain processes to be able to perform the end back task, the same end back task, while in an older brain, it's not the same things you need to inhibit. I mean, if you take a caricatural example, let's say in a 15-year-old, let's say, who is all about something that is hormonally driven, for instance, to perform that task, you would have to inhibit a lot of processes because you want to think of something else. And when you're older, maybe you want to think also of something else. So you have to inhibit other things that you would like to do with your brain. So, and that kind of things could maybe explain why you have a very different signal. So that's just an example. I'm not saying
that's a possibility. But again, how to interpret this notion of inhibition? Is this something that you're voluntarily doing? So say the teenager who's solving this is like constantly thinking about video games and stuff. So whereas the 74-year-old, it's a cartoon, but it's like, oh, this is the most entertaining thing that I'm doing, so there is nothing that I need to inhibit. Then my concern is that if it is a voluntary or a conscious effort to inhibit a process, then you should have something like an introspective report to the fact that there is a difference, and we just don't have them. By contrast, if this notion of inhibition is something at the neural level, then we should be able to see it precisely with the kinds of topological models that I have been suggesting to employ.
Okay, thank you.
Great. I really appreciate it.
Daniel, you're next.
All right. Thanks for a very, very cool talk, Felipe. Obviously, I'm extremely sympathetic to everything that you presented. And as you know, I had things to say about these topics in my publications too.
Yeah.
I just want to say something about the possible objections and replies that you mentioned at the end.
Yeah.
So the first one was these kinds of networks are not explanatory. They're descriptive.
Yeah.
And what was the second one?
So hold on.
It was also something about
the nodes. It's about what
nodes, yeah. And so these two things are interconnected, right? So the reason why people think that they are not explanatory is because nodes are individuated by taste. So nodes are basically arbitrarily chosen.
Yes.
So Craver has argued that, and Woodward recently, that nodes and edges, especially nodes, cannot represent any meaningful working part of a mechanism. And if you can't do that, you don't have an explanation.
Yeah.
So I think having only descriptive networks, I don't see what's the problem with that. I mean, Sarah Green, Bill Bechtel, Arnold Levy have written about non-explanatory uses of networks.
Yeah.
Or exploratory uses of networks. So, for example, it helps you find some part of the causal structure that you wouldn't be able to see or find if you didn't use this particular method, right?
Yeah.
Or it helps you predict stuff without being able to explain them, right? So that's useful in clinical practice too. So what's the deal? I mean, what's the big problem? I mean, so what if they're not explanatory? Sometimes. And well, when it comes to explanatoryness, of course, the reason why people think that they are not explanatory is because of this taste issue thing. And that's particularly raised for the functional connectivity networks, which is, I assume, this is what it is what you presented today. So that's networks that kind of are able to capture temporal dynamics of the system, and you can think of temporal dynamics in many different ways, right?
Yeah.
And the reason for that is because of these time slices and the edges. So the edges between multiple layer networks, or in functional in fMRI, for example, models which are sometimes used to build these networks when you have a task involved, are basically synchronization likelihoods between time series. So it's just probabilistic measure of how synchronized the time series are, right? And so, you know, the objection typically goes from other areas too, that, you know, correlations or synchronization likelihoods or probabilities cannot really, they're only indicative of some underlying stable causal structure that then can, when it's properly filled in or discovered, can actually fill out the mechanism sketch or whatever, something like that. But then the same problem arises for structural connectivity networks that allegedly don't have any of these problems. So they measure, you know, the nodes are brain regions and the edges are white matter tracts, or, you know, direct connections between neurons. But in structural connectivity networks, and so that's what Craver at least said in his 2016 paper. But structural connectivity networks have the same problems as functional connectivity networks because you need the strength
Not
of
only that, if you ever do DTI, and we do a little bit of DTI in our lab as well, there is a number of because what since what so these structural networks that Daniel is talking, for the people in the audience that are not familiar with the method, what they do is that they take advantage of anisotropy, which is a phenomenon in all biological structures in which fluids run in a particular direction. So you can see in which directions the neurons and the bundles go. The problem with that, and so you can see that they're not going in backwards, or they're not going to the sides, although you can actually measure myelin by seeing how they go to the side. That's how they go. But the problem, and I'll let you continue, Daniel, with this very important point, is that sometimes you have to find heuristics to solve process, and everything in the brain is crossed with one another, right? So whenever the two bundles cross, that's tricky, right?
Exactly. And also the strength of the signal and arbitrary thresholds for limiting constraints on data and data sets and everything. So you can turn the table or turn the burden of proof to the people who object in this way. Well, you have the same problem in the structural connectivity networks, which you would probably allow to be explanatory. And so how do you respond to that? I mean, not you, I mean them.
No, I mean, that's a very good point. And it's a discussion that I sort of like, but I think that we as philosophers of neuroscience have just focused so much on explanation. And explanation is only one of the goals of science.
Well,
it's not the only goal of science.
That's true.
For instance, is something that I am very keen on. But that being said, let me just say a couple of things. One of the things that networks, and here's, I guess one first thing that I would say is that I just don't see a single model of explanation in neuroscience, as I just don't see a single model of explanation in many biological sciences, right? So sometimes you can explain things and feel satisfied with an explanation by showing how a particular instance is subsumed under a particular category, right? So why did the person scream, happily when this individual scored a goal, and was very sad when this other individual scored a goal? Well, because he's a fan of the Tottenham, or is a fan of, I don't know, Manchester United, as hard as that can, it hurts me to say it. But right. So those are perfectly good explanations that don't necessarily require causation. And I feel that oftentimes that happens in cognitive neuroscience as well. So we, and especially in network neuroscience. So we have evidence to the effect that individuals with schizophrenia have, I forgot exactly what is the measure, that the topological measure, but suppose that it is more inter-network connectivity in like auditory and verbal areas relative to controls. And then you say, well, what this individual started hearing voices? And say, well, because he has this more, like, again, more interneural connectivity and there's voices relative to others. That seems to be explanatory if you can shift, just locate one individual within another group. And it doesn't sound necessarily too mechanistically, because we often don't know what the mechanisms underlying those networks. But it helps you to, for instance, say, well, then this person might require this drug versus this other person. So that's one first thing that I would say. I think that we just focus way too much in mechanistic explanation, which is a very bona fide kind of explanation in cognitive neuroscience, but there are others in which networks are involved. And the second point that I would like to say is that sometimes explanations and predictions are really hand in hand. Some people would say that, Carl Craver used to say that in his work, that sort of a prediction is kind of like the other side of an explanation. sometimes you can have explanatory power with a topological model without knowing anything whatsoever about the mechanisms that happened before,
Yeah,
right?
yeah, exactly.
So
Mm,
I wrote, yeah, I wrote a paper years ago in which I talked about, for instance, network models to solve traffic patterns in San Francisco, in which they look at the traffic patterns and they know exactly where to start charging more for parking and where to start charging less for parking, and that helped to solve.
mm.
But it didn't matter, like you could be talking about San Francisco or some other
Exactly. It
city.
doesn't depend on the spatial layout of the city, the
Exactly.
spatial instantiation of the network in the city.
Yeah.
It's you get all of it solely from the network.
And you do the same with epidemiological models
Exactly. Exactly. Yeah.
of diseases. It doesn't matter if the disease is, dengue, or if it is what have you. The models help without really knowing exactly what the
Mm,
mechanisms
mm,
are.
mm,
And I think that you have similar kinds of predictability when it comes to networks. Suppose that it turns out that yes, that inter network connectivity is highly predictive of schizophrenia, and then
mm,
it turns out that zapping with transcranial magnetic stimulation, BA22 in the brain, helps to let that local connectivity being less recurrent
mm.
or what have you, and at least make it more like controls, even if for the time being. Then it seems to me perfectly fine to go to the FDA and say, look, this treatment helps reduce the symptoms of schizophrenia. And if the FDA say like, yeah, but what is the mechanistic explanation? Say, I have no idea, but it
Yeah.
works, right?
Yeah.
So
Yeah.
uh, yeah, so those are my views. Not having a mechanistic account of a network or something like that can maybe be bad for one explanatory purpose, but it could be very good for other things that we use science
That's
for.
true. That's true. Actually, I have a paper about how to regiment this primatics of explanatory relevance,
Yeah,
published last year in Philosophy of Science. Actually,
yeah.
I'll
Send it
send
to
it
me.
to you. Yeah.
Okay, thanks, Daniel.
All right.
Yeah,
Thanks.
yeah.
Thank you both. Any other questions or comments or critiques? I don't see any other hands, but especially students, you're much welcome to ask questions, especially those who worked on the paper, but maybe I should have started with students.
Yeah,
Uh,
and the thing was, no, like all of this is complex, and so any question for simple, there are no bad questions, I promise. And if there are bad questions, are the ones I ask. Fabian.
So hello, can you hear me?
Yes, sir, I can. How are you?
So I'm a first year master student in philosophy, and so my question would be, based on this approach, how much inter-individual variation you think we should expect in terms of the way cognitive modules are characterized, and also the generalizability of the structures we may find.
That's a great question. Both of them are really, really great questions. Uh, so one of the great advantages of having a lot of data, and one of the great, so let me just start it. You probably, if you work in cognitive neuroscience, you probably have seen gazillion papers using resting state connectivity. Resting state connectivity is basically the data that you get, or the networks that you get when people are doing nothing, when they're resting in the scanner. Why do we have so many papers with resting state connectivities and networks? Well, the main reason is because everyone that does fMRI collects resting state, right? So I did my postdoc at Harvard, for instance, and when I was a postdoc for 2 years, I scanned basically every day from 7 to 7. It was ridiculous. I was doing nothing but scanning people in the fMRI. And part of our protocol was to include 10 minutes of resting state because they wanted to use the resting state for the center. As a result, in my lifetime there, just by myself, I must have scanned, I don't know, 200 people or so, and resting state of 200 people. I was one of 40 researchers, so we're talking about thousands of data sets of resting state. Um, why is that? Because to be able to get power to find statistical significance, we know we require a lot of subjects. So lots of these resting state connectivities have hundreds of subjects. That gives us, once you have enough power, you have sufficient confidence to the effect that these nodes or these modules are going to be hopefully generalizable to the population you get the sample from. Okay, so that was a long-winded answer to say the power of numbers. How do you know that they are stable across people? Because we have data from a lot of people. Now, of course, when it comes to one specific task, if the task is something that I invented in my lab and I only tested 30 people, I'm not going to be very confident that it is going to be replicable in other samples. But if it is something very basic, like the NBAC task that I was talking, that has been done with thousands and thousands of subjects, and you can generate those maps, then you're going to be more confident. So, so that was the question about sample. And you had a second question, right?
It was, yeah, in terms of the structure and generalizability of the structure, but it's actually linked to this. So
yeah, it's linked to that. So I'm a very austere neuroimager, and I am a very, how can I put it, a very cautious experimentalist, in the sense that I think I try to confine the extent of my evidence or the degree of my belief to the extent of my evidence, and my evidence is always going to be sort of limited to the population that my sample is from. So I wouldn't know if we can generalize this across all humans. I wouldn't know if we can because I don't know to what extent, say, culture or geography or so might have a big influence on this, on the resultant network modularity. So, so I take the evidence with a little bit of a grain of salt, not saying something about perhaps the nature of human brains, but definitely something about the nature of the population of human brains that I am getting my sample from.
Okay, thank you.
All right. Thank you, Fabien, for showing that questions from master students are often the best. We still have 2 minutes. Any other questions? Not that we need another question, but if someone has a pressing question, that's the right moment for that. Fridolin.
Yes. Hello. Can you hear me?
Yes.
I'm Fridolin. I'm from Bordeaux. I'm a philosopher and computational biologist. I don't know this area at all, I would say. And in the beginning, you said you are sometimes a philosopher, sometimes a neuroscientist, and sometimes both. So
Yeah.
is this an instance of where you are both, or could you give us a sense whether the problem or this project is relevant for the neuroscientists or the cognitive scientists as well? And if so, in what contexts?
Oh, I love that question. That's great. So I sometimes I like to say that I am both, and sometimes I like to say that I am neither. So I'm not sure exactly on which field it falls, but definitely I think it's helpful for both. I think it does a job in philosophy in that it tries to get the solution to this parameterization problem, and try to show, that it's very important that we think about that when we think about cognitive systems in humans, that we recognize that we are biological organisms that change over time. And I am very interested in this dynamicity of brains. And insofar as it gives a solution to a problem that was posed in the philosophical literature, well, it's a philosophy project. But also, interestingly, I think it could be helpful in cognitive neuroscience as well. I should say that a lot of what I am lifting here, or that I'm using here, is stuff that Betzel, which is a cognitive neuroscientist and a friend, has employed. So I really like his approach. So I don't think that it is going to be terribly novel for him, and in the sense that they have employed these kinds of structures and these kinds of mathematical parameters to solve their own problems. What might be helpful for him is to show how this could have further implications for the kinds of things that they want to say. Betzel and colleagues normally use parameters, the time parameters that are very small, in the order of, seconds, or maybe they learn a task and then they test them minutes after and so forth. And what I am trying to say is that you can actually extend this this sort of studies to temporal dimensions, or temporal windows that are longer, right? If you, as long as you can control for change. So I had another, in progress paper to further develop that omega parameter when you have evidence of decay. So for instance, just to give you an example, white matter decays over time because white matter is basically myelin, which is this kinds of cells are called Schwann cells. And at the age of 33 or so, we stop generating them because our body said, like, no more. I mean, we normally just live 60 years. That's why motor control starts to decay if you live, over 70 and so forth. So those biological parameters should be taken into account when thinking about that omega parameter in the big formula. So I think that is an extension that could be helpful potentially to neuroscientists who are interested in employing these kinds of solutions to temporal windows that are longer than the order of TRs or milliseconds or seconds or minutes.
Thank you.
Yep.
Thank you very much. We're perfectly on time. I really want to thank you again, Felipe. That was great. Thank you so much. Thank you for the questions. And I really hope that our path would cross in person in the future, Felipe, because it was really a great experience to hear you today.
Okay, well, thank you so much, everyone. Thank you, Thomas, for inviting me. And I hope you all have a wonderful day.
You too. Bye-bye. Thank you again.